Okay, let's continue the discussion. Okay, C. So students found that compressed yeast gave highest number of, hang on, something pop up. Okay. So students carry out 2 further experiments to find out the best conditions for the growth of compressed yeast. So the students want to find out in the experiment what is the best condition for the yeast to grow. So the student did experiment to find out two variables here. The pH is the independent variable, as well as the incubation time is also the independent variable. So absorbance here has to do with continue from what we learned last week, whereby when yeast respire, so respiration, okay, whether it's aerobic, anaerobic, okay, it will lead to a lot of dehydrogenation reaction, which and therefore will give rise to a lot of H2 hydrogen atoms per dehydrogenation reaction, and then this will be carried by electron carriers. So now in this experiment, we are using TTC as an alternative or as a substitute to the electron carriers. So the electron carriers we are talking about here is NAD plus and also FAD plus. Okay. So when TTC accepts the hydrogen, the oxidized version will become reduced TTC, and hence the color will change from colorless to become red color. Okay, and then we're going to use the colorimeter to read the intensity of the red color, okay, by either reading either the absorbance or the percentage transmission. So either percentage absorbance means how much of the light is being absorbed by the red particle. So the more the reduced TTC, so the more light is absorbed. Okay, so the lower is the percentage transmission. Okay, so therefore now this is the reading. The absorbance, so they read absorbance means how much of the TTC is produced. The more the TTC, the more the absorbance in arbitrary unit. Okay, so you can see that among all the points here, the highest point is actually this point here. So let's look at the condition for the highest point where there's most absorbance, and the most absorbance, the most the TTC is being produced, the highest the rate of reaction in terms of per hour. Okay, sorry, not per hour, in terms of a highest amount of product produced in a fixed time. So this is pH 7.5 at 4 hours, the rate is the highest. And you look at this bar here. So they say that this is the standard error. Standard error of this one does not overlap with the rest of the standard error at 4 hours. It means this is significant. The differences between these 3 pH at this time and the rest of them, they are all not overlapping. This does not overlap with the rest. So it is significantly different from the rest of them. Okay. So now we go on to the question, what the question asks the student. This particular paper 5 got a lot of statistical question, a lot of statistics, right? It's like everything you learn, they ask you. So part 1, state what the standard error shows. So very easy. The mark scheme answer is the standard answer, which is the first one. The first one here is the standard answer. Standard error shows us how close is the sample mean to the true population mean. That means your mean value is a population mean, and 95% of the chance, okay, if you obtain your data from the same population. So this is a population, right? You already obtained population mean from here. There's a mean value for this one. Okay. And then if you also take the data from the same population, 95% of your data, as well as 95% of the chance when you get a data, it will be found, uh, close to this population mean. Okay, 95% of your data will be found close to the population mean. So this is the standard error, the meaning of standard error. How close is the mean that you obtain from the data is to the true population mean. Now, the red color one is the one I've added in. It's an alternative version of the meaning of standard error. So when to use this is you need to look at the scenario in the question, right? Whether they've given you the population mean, and when you conduct an experiment, you're trying to obtain your sample mean, or whether in your experiment you obtain sample mean, you want to predict the population mean. Okay, so that you need to know which one to use. So the standard one is the first one on top here. This is a standard definition of standard error, and the rest are alternative, depending on scenario. How close is the true population mean to the sample mean? So you take a sample from there, you want to predict your population mean with 95% confidence. Or you have taken the sample mean, you want to know the second sample mean. How close is the second sample mean is to the first sample mean? Okay, so that is called standard error. So it's plus minus 2 SM. Okay. So number 2, the graph figure 1.3 shows the 95% confidence interval. I like this term. So we need to revise again the meaning of this term. What do you mean by 95% confidence interval? In all the statistical tests that you learn in A2, statistical analysis, all of them make analysis based on the 95% confidence interval. That means to say that if you take the whole class to conduct experiment, what will be the range of the result that you'll get for 95% of the students' data? So this is called 95% confidence interval. Okay, so what does this mean? 95% confidence interval, part 2. Okay, that means 95% of the data that we obtain would be expected to lie within this range. So it is same as the whole class do the experiment, 95% of the students' result will be expected to lie within this range. Okay, because this is how we get the 95% confidence interval, because whatever test that you've learned earlier on, we say that if you plot the graph, okay, for example, the graph of what? The graph of, what is this? The graph of chi-square value. So the whole class get conduct experiment, and then you calculate the chi-square value. You expect to see that the chi-square value of the class data will go down this way, right? And then the area below the curve represents 100% of students' data, and we want to have a confidence interval of 95. That means this is called the extreme chi-square value that is very far from the expected value here. Okay, so therefore we reject all these value here and retain the 95% confidence interval. That means 95% of the data will lie within this range. Okay, and because we reject the extreme 5% N. So that gives us 95% confidence to make conclusion. Okay. So this is the first marking point. Second marking point is, what do you mean by the range? Okay, see. What this indicate about the data is what this 95% confidence interval indicate about the data. The first marking point is giving us the meaning of the word 95% confidence interval. Okay, 95% of data will lie within this range. And the second meaning, according to the question in figure 1.3, so we go to figure 1.3 and look at what does it mean when they show you the range. That's why that's the second marking point. First marking point is the definition. Second marking point is based on the data, right? So figure 1.3. So all these are the confidence interval, 95% confidence interval. What does it tell us about it? They tell us that at 4 hours, right, for all the 3 pHs that we use, the confidence, 95% confidence interval, they do not overlap. So when they do not overlap, their differences are significant. That means if you use different pH, then the absorbance that we get will be different. That means the rate of respiration will be different. So it's significantly different. So that's the answer for number 2. Okay. At 1, 3, 4 hours, the sample mean was reliable, or there is a significant difference among the means for different pH that we use, right, or different hours that we use. So 1 or 3 or 4 means because the question said from the data from the graph, so we need to quote the value from the graph. So at 1 hour, all the 3 interval confidence interval level do not overlap. All these 3 do not overlap. All these 3 do not overlap. Only at 2 hours, you can say that they actually overlap. So therefore, the answer is at hour number 1, incubation hour 1, 3, and 4, the 95% confidence interval do not overlap. So they are significantly different from each other. So that's where majority of students wrote, right? Okay. If you write reliable also can, it means when you keep on repeating experiment, you will get your data within that 95% confidence interval. But this is not how we learn. We learn that they do not overlap, means there's significant difference. Okay. C, part 3. Okay. After completing these 2 experiments, the student concluded that the growth rate of yeast is highest when incubated at 30 degrees Celsius and pH 6 for 4 hours. So that means you need to base your answer based on these 2 experiments. The first one, they give the graph. The second one, they give the table. Okay, so the table is they redo the experiment and they now use different independent variable to test again. So now they use different temperature. Just now in the graph, they use 30 degrees Celsius only. So over here, they want to test other than 30 degrees Celsius, any other temperature that give higher rate than 30 degrees Celsius. And the rest of them, they fix 4 hours. Okay, they fix the time as 4 hours because in the first experiment, 4 hours give us the highest rate. So we choose 4 hours and repeat experiment. Now we test the temperature. And we also test again the same 3 pH. Okay, so this second experiment, so we got first and second experiment. What they want the question the student to answer, state 2 ways in which the data support this conclusion. The growth rate of the yeast is highest when incubated at 30 degrees Celsius, pH 6.0 for 4 hours. So when you want to use your data to support the conclusion, that means the data give us mean value. So you must look for the highest mean value for absorbance. Absorbance, this is number 1. And then in order for the conclusion to be valid, we must include the result from statistical analysis. So that will give us 2 marks. First mark is from the table and from the graph, look for the highest mean absorbance. Second mark is you must verify whether this mean that's highest has got a 95% confidence interval that does not overlap with the rest of the results that you obtain. So only using these 2 that your conclusion is valid. The conclusion is valid. You cannot validate unless you got statistical analysis. First, you must have mean value to show which is highest and lowest. So marking point says that, so we start from the graph. So from the graph, okay, it shows that pH 6 will give the highest absorbance at 4 hours incubation. Okay, pH 6, and also you can quote that the highest absorbance is 3.16 AU at pH 6. Okay, so this is pH 6 here. This one cross pH 6 at 4 hours has got the highest absorbance, and you read off the value is 3.2, right? 3.16 AU. Okay, or so first marking point is code data. Either from the graph or from the table, they only give one Is it 1 mark? This or this. That means either from the table or graph, they only give 1 mark for code figures. So from the table, at 30 degrees Celsius, pH 6 absorbance is the highest. So table got many temperatures, so if you choose 30 degrees Celsius, give you the highest absorbance among the rest of the data. So 30 degrees Celsius at pH 6. Okay, it gives the highest absorbance. Then what about statistical analysis? So statistical analysis part, then you must say that we use statistical analysis. The statistical analysis is based on the standard error. Okay, standard error. Oh, sorry, the error bar plus minus 2 SM. Okay, it's based on confidence interval, which is actually the error bar. The error bar. Okay. What else do we have other than this? Let me see. Or standard error is the same, right? They're all the same. Standard error do not overlap, it's the same. Standard error, the error bar is mean plus minus standard error. Mean plus minus 2 standard error. Okay. And this is also confidence interval. I noticed that some students, when they write the value, right, when they quote figures from the graph, they didn't put the unit, especially when it comes to AU. AU, artificial unit. Okay. Okay. Now, question number 2. So let's go to question number 2. So question number 2 is about ecological sampling. So again, it's statistical analysis, a lot of calculations. So let's look at the story and understand the story behind the question. So they say that this white clover is a kind of plant, and it is trifolium repens. They're described as cyanogenic because they can produce hydrogen cyanide. So this plant can produce hydrogen cyanide, which is a poison. And this poison inhibits respiration, means it could have inhibited enzymes that are used in respiration. And then because of this, it actually deters the herbivores from grazing onto the plant. So this is a way to protect the plant so that when herbivore eat the plant, right, the respiration rate of the herbivore will be affected. The herbivore becomes weak, for example. Or if it's serious, they may even die. So they will try to avoid this plant because when they become weak, that means they feel unwell. So they know that they cannot consume this plant. Okay. So a group of scientists investigated the percentage of cyanogenic plants in population of tea repens growing in different locations. So they choose January at different temperatures to find out the population density of the plant at different temperatures in different places, okay, in the country, no, in different countries, okay, in different countries. So all of them has got the same what? Month. So the experiment is conducted at the same month, and when they measure the temperature at the month where they conduct the experiment, these are the range of temperature obtained, okay? And this will be the range of the percentage of the cyanogenic tea repens. Okay, so by now you know how you calculate the percentage, right? You can be calculating percentage cover using the quadrat. Okay, that is the one that you learned only. Okay, percentage cover. Okay, so question. So the scientists calculated Spearman's rank correlation. So we try to look at the correlation coefficient between the temperature and the percentage cover of the plant. Okay, so state the null hypothesis for this test. So null hypothesis is always there's no significant difference. In this case, it's not difference, it's correlation. So there's no significant correlation between the temperature and the percentage cover of the plant. A learning point from this question is that when you want to write the hypothesis for statistical analysis, please include the word significant. There is no significant difference about what? No significant difference, no significant correlation, no significant difference between observed and expected value, no significant difference between mean of data A and data B. Okay, so no significant what, right? And it's underlined means you need to write this word to score. So that's where a lot of students didn't get marked because of that. Okay? Because of not writing the word significant. Okay, B part 1. So the formula is given, so you're asked to actually calculate, right? So number 1, you're asked to rank, right? This is the percentage plants, and this is the temperature. So you got a pair of data, and they ask you to rank all the individual data, okay, for both the variables. So therefore, the answer already given to you. So we always rank from the smallest number to the biggest number. So smallest number, 0 is 1. Then followed by the next higher number. 5 is 2, and so on. Same as for the temperature, lowest is negative 23.9 is 1, and highest is 12.2. Okay, it's 8. So this is how you rank it. And the calculation is very logically, you can see here that, okay, I'm going to calculate the difference between the two ranks. It doesn't matter whether you take 8 minus 8 here, sorry, when you take 2 minus 1 or 1 minus 2, it doesn't matter because ultimately you're going to square them. The moment you square them, you remove all the negative value already. So it doesn't matter whether you take A minus B or B minus A. Okay, so here you can see that take the first value minus the second value and then square them and sum them up. So summation of D square, and then you use the formula that's given to you to calculate the correlation coefficient. Spearman's correlation coefficient, and the formula is already given earlier on here. So this one, So now you got the D square value already. You want to know what is N. So N means total number of paired samples obtained. So just count here, 2, 4, 6, 8, 9. Total number N equals to 9. N is the number of pairs of items. So this one is very straightforward. All the calculations, most of the students get it correct. Most of students, which is very good, right? So they even ask you to put your answer in 2 decimal place, and in the answer they purposely write here 2 dp, do 2 decimal place. Okay, so the table 2.3 shows critical value. So this is critical value. That means all these values here are not RS value, they are critical RS value. Okay, at the 0.05 probability level. 0.05 probability level means when you get your RS value at this particular value, the probability of variable A correlated with variable B is significant. 0.05 means significant. Okay. So the calculated RS value is what value here? 0.88. Okay, calculated RS value is 0.88, and the total number of sample is actually 9. So let's look at the value in the table. N is a total number of sample is 9. So this is the one we are supposed to look at. 0.68 is the critical RS value. So our calculated RS value where is 0.88. Okay, so our RS value that's calculated is equals to 0.88, and this 0.88, okay, this is how I'm going to write it. The RS value bracket 0.88 is actually bigger than critical RS value, and I'm going to put in bracket 0.68, like this. So write minimum, score maximum. So what does it mean? That means to say that the null hypothesis is rejected, okay, because the RS value is higher than that. So reject null hypothesis. What does it mean? That means there is a correlation. There is a correlation, okay, between the variable A, which is the temperature, and the variable B, which is the percentage cover of the plant. Okay. So calculated value 0.88 is greater than critical value 0.68. Okay, 1 mark. Okay, I like this question, thinking question. Suggest why there's a higher proportion of synergic clover plant at the location with mean January temperatures compared with other temperatures. What mean January temperatures? What is that? Mean what? Higher? Why there's a high proportion of scientific level at locations with high mean value? Okay, so when the temperature is higher, why the proportion of the plants is higher? So when it's higher temperature, the plants grow better. So when the plants grow better, of course, it has got higher density. But some students say like this, some students say that I want you to write and show you higher temperatures means higher photosynthesis rate. I cannot give mark because photosynthesis rate, right, is basically mainly affected by light intensity. So even if your temperature is very high, but there's no light, photosynthesis cannot occur. So therefore, because of that, I cannot give mark for students who say, oh, higher temperature, so the rate of photosynthesis is higher. Not necessary. So when it's half correct, half wrong, the answer will be wrong. So the only way you can write is that plant can grow better at higher temperature. Why? Because you're not talking about photosynthesis, you're talking about growth rate. Okay, growth rate, temperature higher, so therefore the rate of reaction becomes higher, right? Kinetic energy is higher, effective collision is higher, ES complex form is higher, therefore the rate of synthesis of component of the cell, biological molecules becomes higher, so the plant will grow faster, like that. Okay, rate of metabolism becomes higher in terms of synthesis of carbohydrate, lipids, and protein for the components of the cell like membrane organelles, okay, cytoskeleton, cell wall, and this on, and therefore growth of cell. Okay, so that is the first part. So this is where you got to be very careful. What leads to what? Okay, light leads to photosynthesis, temperature leads to higher rate of reaction, not necessarily photosynthesis. Okay, even though it can affect enzyme activity also, right? So also, because they grow faster, they might produce more hydrogen cyanide, and hence they are able to deter or reduce grazing or kill more herbivores, because when the concentration is higher, when a herbivore feed on it, it might become poisonous and then herbivores may die, or it may actually deter the herbivores from eating them. Right? So therefore, they are able to survive grazing better. That means herbivores don't really graze on them. They survive grazing better. Okay? So this is question number 2. So now we go to question number 3. Yeah? So question number 3 is ecological sampling of pig box. Right? 2 pig box, pig box ecosystem. A and B. Yeah? So they said pig bog are wetland. That means this place has got certain location that is immersed in water. And hence, you got different kind of plants growing there that's suitable to grow in the water and around the water. Okay? It's a type of wetland covered with water. So pit bog. Okay? So, what is the question? So the ecologist sampled plant species in pit bog A and B, and they decided to use 2 suitable indices of biodiversity. Oh, I like this question very much. Okay, so we have learned Simpson's index of diversity, but now they introduced to you there's another type of index that can be used to measure biodiversity. Okay, and then you learn that biodiversity can be studied in 3 levels, right? Ecosystem diversity, species diversity, and genetic diversity. And then in terms of species diversity, there are 2 things we can study, right? Species diversity, right? So there are 2 things we can measure there. Number 1 is species, the evenness and species what? Species what? Number 1 and number 2. Number 2 is species evenness. And number 1 is species what? Abundance. Abundance. Okay. Different number of type of species. Number 1, different number of type species. Number 2, total number of individual of a species. Total number of individual of each species. So this is from theory what we have learned about species diversity, and this particular experiment is actually finding out species diversity using species diversity index. But then we do not know the second index. What is the formula? What do they what do they what do you call that? Emphasize. Different index emphasize different thing. Okay. So the ecologists had read the two indices and they know that it can lead to different conclusion because each indices, they emphasize different components of the species. Okay, so the Shannon Diversity Index gives value in the range of 1.5 to 3.5, where 1.5 is low biodiversity, 3.5 is high biodiversity. So this is something that they just give you information. So let's look at the data here. Okay, so let's make it smaller so we can see everything in one. Okay, can. So Simpson's index of diversity for pit bog A is lower than pit bog B. But Shannon's index of diversity is higher in pit bog A than pit bog B. So let's put this value here. So this is lower, but this is opposite. This is higher. So both are index for biodiversity or the species diversity, but how come they show different conclusion? So the question asks a student to deduce why is it they lead to different conclusion. Let's look at the phrase of the question. Okay. Where? Okay. Suggest how the differences in the data in two samples shown in the table may have led to different conclusion. So keyword, how the differences in the data give to different conclusion. So these are the two keywords you want to pay attention to when you answer the question. Talk about differences in the data that you can see based on the theory that you have learned. That means when I study species diversity, I pay attention to species abundance and species evenness. Abundance means different number of species. Evenness means different number of individuals of each species. But in this case, it could be different number of individual in what? One species or the entire, all the species add up together. So you have to see. Okay. So this is in terms of differences in the data. This is what we can use our theory to apply to answer the question. But then don't forget about this keyword, different conclusions is where a lot of students didn't get the mark unless they have done this past year. Some students, right, why they score very high here? Because they have done the past year, they memorize the maxim and write down maxim answer. I know. When I mark, I know. They give me perfect mark scheme answer. Okay? Right. So what if you're not done this question, how you can still score full marks? Read the question and look at the keywords of the question and answer the keywords. Because they didn't conclude, you know. See, this is Simpson's index, this is Shannon index, right? But you see that the conclusion there didn't say which index. One index indicate pit bog A had more biodiversity, and other index indicate pit bog B has got more biodiversity. So they didn't tell us which index tell us that pit bog A has higher, whereas which index tell us that pit bog B has higher biodiversity. So we need to give the different conclusion. Apart from what is the difference in data that give the conclusion. So that's where you can get the full marks. Okay? So students who get the full marks, majority of them, they've done the past year. Okay? So 2 marks, So let's look at the 2 marks. So this is the first marking point. The table is alternative way of expressing the same thing. So pit bog A has got higher number of species. So this is called species abundance. Okay? So therefore, which index focuses on or place emphasis on this to give them the value of biodiversity? Shannon's index of diversity pay attention to the number of species. So therefore, you see that in which particular data got higher number of species, where? Here. Okay? So this data, wait, higher number of species, yeah? Now, this one here is totally zero, right? Okay. Whereas this one here, pig bog A, all TUVW. So they got these 4 species, additional species that the pig bog B does not have. And Shannon's index is higher in A than B. So we say that Shannon's index actually measure the total number of different species, okay, which is called species abundance. So look at the mark schema. Higher number of species. So pig-bog A has higher number of species, and therefore it gives the higher Shannon index of diversity. Pig-bog B has a smaller range of species abundance. That means the total number of different species are less, and it's got a higher Simpson's index of diversity. I disagree with the answer. I don't like the answer. I prefer the answer to be pig-bog B has a higher, this is a better answer. Pig-bog B has a higher total number of individuals, and therefore it's got higher Simpson's index of diversity. It means it pay attention more onto total number of individuals of all the species added up. Okay? If you write species evenness, you will also get the mark. Species evenness. Okay? Even if you talk about individual species, the number is actually higher. Okay? You see, this is 24, this is only 15. This is 16, this is 8. All of them are higher. See not? If you compare between different species, all of them are higher than pig-bog A. This also higher, this also higher, except for one. This is also higher. All of them are higher. So individually, they're higher. You add up total number of different species is higher. Each species is also higher in number, except for L. This is 30, but this is 34. Okay, but the rest of them, each species, their number is higher in B than A. So it applies whether you say individual species or you say total number of species. Add up to give you total number of individuals is the highest. Okay. So that's the last question. I think I've completed answering. Got some more? No more, no more. One more, B. Okay, one more question, B. Last question. Describe how this sampling method could be improved to make sure that the results are representative of all invertebrates in the whole pig-bog A. So they tell you the story of how this experiment is conducted. So they give you, okay, these are the criteria where they conduct the experiment. Okay, where is it? Here, right? So they ask you for each of this item here, how can you improve this experiment by improving these variables that have been used in the experiment? So very easy. Most of you can actually get the marks. Okay. So you see the timing that they use is between 1 to 4 p.m. So to collect the samples of what? Of the invertebrates. But you know that some insects are nocturnal. They only come out at night. So if the student carry out sampling in the afternoon, they may miss some of the insects that are nocturnal. So therefore, this experiment is not inclusive, right? There'll be error in experiment. So therefore, also sample during the night time or different time of the days other than 1 to 4 p.m. And they also said 3 different days within 30 days. It means they only make the sampling in one particular month of the year. Now, in a country with 4 seasons, right? The temperature and the condition, the rainfall will be different at different seasons. So you want to sample in different season. Even like in Malaysia, you've got rainy season and dry, not really dry season, but you've got monsoon season when there's more rain. Okay, non-monsoon season is less rain. Whereas in Thailand, they got very obvious rainy season and dry season. So you want to include those seasons there. Okay. And then they said they sample along the edge of the path. So that means to say that there's a path, and then to make life easier, the student just walk along the path, okay, along the pig bog, and then how come it's gone already? Walk along the path in the pig bog, okay, and then they just randomly put the quadrat, okay, put one here, maybe put one here, put one here, put one there, finish, okay, put one here. like this. So they only sample along the path, which is actually not inclusive of the entire area. So we need to randomly sample a bigger area rather than just along the path. Okay, and they use the 1 meter square quadrat. Now, when I teach you this chapter, right, I give you two other ways of sampling insects, right? Because insects can move around. You put the quadrat there, right? And then you go and sample them. You go there, they jump away. Because insects can move, they can fly away. So another way for us to trap the insects is we can use putter, right? It's a bottle where you see the insects, you suck them in. Or better still, you use another method, which I call it the pitfall trap. Pitfall trap means you dig a hole and you put a drum into the hole there. You cover it on top so that it's not subjected to sun and rain. And then the insects will fall into it. Insects that pass by during that range of time will fall into it. Then you might be able to open up the cover and then start to observe the insects and then identify them and count their numbers, right? Take photograph and then count the numbers. Now, also they mentioned about last one, they use identification key to identify the species. So nowadays, there are many, many ways to identify the keys. If you look at the mark scheme, it's very interesting, okay? Because they say that you can even use the apps, right? Where's the apps thingy? Okay, identification apps in the handphone, isn't it? There are a lot of apps. You take the photo and then you post it up even to the Google, right? They can actually tell you what it is, isn't it? Okay, Google Shoot, is it? Okay, so you can use apps or you can get an expert. So you take a photo and then go back to your science lab and there's an expert, insect expert, which can actually look at the photograph and tell you what species it is, right? You can use expert, you can use identity app. So there are many, many ways to double check that is correct, like in the sense. So I will say that this is to double check. Use two methods to double check. Okay. So that's all for today. Stop.